HPC division at the Blue Brain project. He leads the development of the circuit building software stack to establish the neural connectivity of digitally reconstructed brain regions and to maintain scalability for future scientific endeavors. To this end, Matthias is working with both MPI as well as modern data analysis frameworks like Apache Spark. Before joining the Blue Brain project, Matthias concluded his PhD study in high energy physics at the University of Notre Dame. So today he will be presenting on building brain circuits, experiences with shuffling terabytes of data over API. So let's welcome Matthias, and you can share your screen and please get started.
Hi, thanks. I hope you can hear me.
Yes, we can hear you, but we are still waiting for the slides.
Yes, sorry, I was just trying to find out the right window. Okay, so now you should have my slides. Okay, thanks for the introduction. So I'm Matthias. I'm a big data software engineer at the Blue Brain project of EPFL. So if you attended last year, you may have seen Pramod Kumbar's presentation on doing actual brain simulations at the Blue Brain project using Neuron that we've just seen mentioned as a benchmark in the previous presentation. And I want to here give an overview over the step beforehand, not doing the brain simulation, but actually building the connectivity we use to simulate the brain. And in the process of doing so, we ended up shuffling quite a bit of data around. So with that, just want to give an overview of the structure of the talk. So I'm going to dive down somewhat in the background. Then we're going to go in and briefly discuss how we actually built these brain circuits. Then I'm going to talk about the corner piece, the software. That's a piece of software called Touch Detector. Briefly discuss the algorithm and the some implementation details, to then extract the centerpiece and talk a little bit about benchmarking MPI of that. And then I'm going to give a brief outlook. So, yeah, part of EPFL. So EPFL is one of two federal universities in Switzerland. So the other one you might have heard of more is ETH Zurich. It's in the German-speaking part of Switzerland. EPFL is in Lausanne in the French-speaking part. Yeah, it's situated nicely on Lake Geneva with a good view of the Alps. This here is the Rolex building. It's kind of a library. And it has about 11,000 students and about 5,000 academic staff. So our project moved from Lausanne down to Geneva at the southern end of Lake Geneva a couple of years back. So the Blue Brain project was started in 2005 to explore brain simulations. And it's located here at Campus Biotech in Geneva, which is a hub for life sciences. So what do we actually want to do in the Blue Brain project? So we want to basically move brain science into in silico based just on simulation. And for that, we mostly focus on rodents, such as the rat and mouse, whose structure is pretty close to the human brain, but it is way less dense, so it's less taxing. Because to jump to the human brain, we'd have to go up by about 3 orders of magnitude in complexity. And we set the initial groundwork somewhere between 2005 when we started in 2010 with a column of about 10,000 cells that you could actually build the brain structure on a desktop computer. And more recently, we've scaled up from and a one column is what we call with about 160,000 cells in 2015, all the way up to the rat neocortex in 2019, which has 10 million cells. So on top of the complexity of the sheer number of cells that we want to simulate, we also have added complexity from the difference of scales we can look at. So on the left side here, we have a point neuron model, which is basically every nerve cell is just one point in space, and we have single connections between the nerve cells. So that makes simulations pretty easy. That makes the brain building pretty easy. You just place a neuron somewhere and you connect them statistically. Then if you just look at the one cell and you expand it out into its structure, it has a tree-like structure with a lot of branching, like this here in the center. That's a morphologically detailed neuron. And then you basically want to establish the connectivity between the neurons by how these branches touch. And then to make things a little bit more complex, you can say this level of detail is still not enough, and then you zoom in and look at the single compartments of the neuron cells, of the nerve cells, and you basically model the flow of the chemicals through the compartments. And so that's the subcellular or molecular level. So, and for us at the Blue Brain Project, we mostly focus on detailed neurons with electrical or chemical transmission. Our approach is basically that we kind of reverse engineer everything. So we take a lot of input data from labs. We try to abstract it away into a conceptual model. Then we take this conceptual model and expand it again into a computational model of the brain. We run our simulations on it, and then we go back and compare with the biophysical model, and we see where the differences are, where we maybe need more data, or where we need to adjust parameters we assumed. And so here basically I want to talk about going from the conceptual model to something that is ready to simulate. So how do we do this, or where do we do this better? So we have our own supercomputer. It used to be Blue Jeans up to this iterations. This is located at CSCS in Lugano, which is at the very southern end of Switzerland. It's in the Italian-speaking part. We started out with what you see here with these racks. Those are about 200 Skylake nodes, dual socket. We have recently expanded and added up to 800 to 900 Cascade Lake nodes. These are connected with 100 gigabit InfiniBand, fat tree structure. We have some additional GPU nodes. Those are more for machine learning. About 6 petabytes of storage in GPFS, which we make good use of. And we have an additional burst buffer based on IMD technology. So this machine was built for us by HPE. So naturally, we run HPE MPI on it. But we also like to investigate MBA pitch as an alternative. So we kind of avoid a little bit of a vendor lock-in, and we also are trying to eke out a little bit more performance. It should also be noted that MBA pitch was faster than HPE MPI to integrate IME support. Okay, so back to brain circuits. So how do we actually go get the base data for these brain circuits? So for a whole bunch of years before we started the project even, what we've done is that a rat here on the bottom right has been so kind and donated us its brain, more or less voluntarily. Then we take this brain and we slice it in the wet lab. And then we have basically take two approaches. One of them is that we stick it into an apparatus for patch clamps. These are very fine probes that can be made attached to a neuron at some point. And then you have a number of them. So you get, you attach to different points of the neuron, you stimulate one of them, and then you record the response on the other ones. And so this gives us kind of these response curves you see in the upper left. This is basically part of the classification of neurons and also used by the simulator to actually do the spike response of the virtual cells. And the other approach is the other data we extract, which you see here in the center, is that we go in with a microscope and then we trace this neuron, and then we basically get this nested tree structure for the neurons. So then once we have this data, we don't actually collect enough data. This would take a lot of years and manpower to do, to assign each neuron individually one shape and one response. So what we do is basically we vary these shapes and responses a little bit to get a broader statistical sample. Then we take a brain volume we want to model. We take the biologically expected distribution of neuron types, and we place our cloned data into it. And then as a step that I want to discuss here, once we have this 3D volume populated with neurons, we need to establish where these neurons are close by and should form synapses. And this, for example, is how it looks like, what the circuit looks like for 10 million neurons in the neocortex. And just give you a ballpark, we have about 150 billion synapses in here. So that's 150 times that one neuron came close to another. And if you want to simulate that with Neuron, we need about 170 terabytes in memory. So the centerpiece to detect the connectivity between neurons is called touch detector. And yeah, we use a lot of distributed computing for that. First of all, I want to go back into the nomenclature just to prepare you for the next couple of slides. So yeah, as you might have gathered, a neuron is just a nerve cell. Then whenever a morphology comes up, that's basically just the physical shape of the neuron, so how the tree structure looks like. And there we have a central part, a soma, that's represented here by black dots. And then we have branches, mainly axons and dendrites. And as you can see here on the right, we have them kind of color-coded. We basically have these blue branches of the axon, and they touch the dendrites, and there's a directionality to all these connections. So we have one side that is emitting touches and the other one that is on the receiving end. And when I talk about touch, I more mean like this is a physical region of the neuron where it is close to another one. And then when we talk about synapses, this is more a punctual connection. So in biology, this would be like a small tube that stretches from one part of the neuron to another part of the other neuron. Okay, so with that, how does the actual calculation we do look like? So as said, these touches are directional. So basically, we have one emitting part of a neuron and one receiving part of the neuron. We model both just as a sequence of cylinders. And then, yeah, we just have a cylinder-cylinder overlap detection in 3D space. And all the complexity just comes from that this has to be done for 10 million neurons of varying complexity. So depending on the cell model you use, there can be hundreds of branches and thousands of cylinders per neuron. So, and this overlap is basically, just to drive it back home again, saved as an overlap, as a projection on that cylinder axis. So it's a spatial region. Once we have all the overlaps between two neurons, we take the spatial overlap, the region, and to kind of homogenize, because these cylinders could have varying lengths. If the cylinders have varying lengths, there might be, for the same density of cylinders in one volume, we might have a different number of touches based on if the cylinders are larger or smaller. So we kind of homogenize this by, see here in figure A, we take these regions, if they're close enough, we merge them into one gigantic one, and then we have some biological data as input, and we space out punctual touches and then connect the punctual touches to the counterparts of these regions from figure A. Yes, and this last step basically requires that wherever we did the cylinder-cylinder overlap, we communicate back to one rank all the results to do this redistribution. So when we scale up, this is a kind of simple process actually. First, every rank loads a whole set of neurons. Then we take the coordinates of the center of neurons or the centers of branches, because taking all the cylindrical coordinates would be a little bit too much memory to sort. Then we partition the space of the brain up such that every rank will be assigned a rectangular region of the brain of about equal density. So in this case here on the right, this is about 2 neurons. Once we've established this partitioning, we will do an all-to-all to exchange the neuron data so that every rank has the data assigned to it, and then it'll build its own spatial index of that data. As I mentioned before, these are directional touches. So we do this only for the postsynaptic, so the receiving side of the data. And in the actual processing then, we take the other side of the neurons, we load it in batches, because once we do this overlap detection, we generate a lot of data, so we don't have enough memory to keep everything in memory at the same time. So we load a batch here represented by just a singular neuron. On any rank we do this. Then we find out where in the space partitioning it overlaps with different ranks. We distribute the neuron data to the ranks which do the overlap. And then finally, at some point, we will be sent back the results, and we do this post-processing, and then we write out to disk. And while we basically wait for other ranks to process the data we send out, we process the data that other ranks send us. So I think if you were to graph this out, that would be really interwoven. I should also mention that we've simplified our process recently. We've added some one-sided communication, mainly to keep track of offsets when writing the files, and to keep track of which neurons we actually have processed. So to summarize, basically, we kind of have a two-step algorithm here. First, we load the postsynaptic side. We create a distributed spatial index across all ranks. And then every rank in the second phase, which I call here the work phase, every rank loads a batch of neurons, distributes it to remote ranks, collects overlap data, redistributes, and writes to disk. And it keeps repeating until we've processed all neurons. And so the communication patterns we use here are basically split along the phases. So at first, we do a lot of collective operations, so all reduce, all gather, all to all, and the vector variants. And then finally, in the second phase, we do a lot of asynchronous sends, a lot of probing, some file writing, and a bunch of window locking, unlocking, and fetching up for the one-sided communication. So here, we transfer a lot of data, not so much in the first phase, although I think for an all-to-all, this is still pretty big. So the spatial index for 10 million neurons is about 2 terabytes total, so 500 megabytes per rank that we send over the network. That takes a little bit of time. It's not too problematic because we only do this once. And over the whole work phase here, where we iterate over these batches of neurons, we send a total of about 60 terabytes for this circuit from one rank to other rank, between all the ranks. And that works out to be an average message size of about 100 to 200 kilobytes per rank-to-rank communication. And then the data we collect is actually quite rich. So that's 100 terabytes we receive back before we simplify the data and write it out to disk. And so here, should note that this is all asynchronous. This is all varying inputs. So all the neurons have different sizes, different locations. So that means that basically the communication pattern changes. There's a varying amount of ranks involved in every communication, and also varying amounts of data sent out to every rank. So at some point, we decided we would like to profile this. The software is actually quite old, so we've maintained it. We decided to profile it a bit to see if with our new machine we could eke out some more performance. And we discovered that actually MPI is taking a lot of time. So so here's the MPI volume pattern too. So this is really like just showing that we don't have a lot of volume, we don't have quite a pattern in here. It's mostly, equal but kind of randomized communication. So we abstracted the design of the algorithm a little bit to be able to investigate what we could do to tune our MPI performance. And so basically, we just want to focus on the part that consumes the most time, which is the sending data out to overlapping ranks to do this remote work and then collecting back the results. As I said before, the amount of ranks involved varies. It varies mostly with the problem size because we do this at all kinds of scales, and it also varies with how big of an allocation you run on. So we basically built a mini app for this. We removed all input data dependence, we removed these one-sided calls for bookkeeping, and we basically simplified it to sending just data out and doing no computations. I think there's a small sleep in there just to simulate something, but it's pretty homogeneous actually compared to the heterogeneous work we do. And here, basically then, the parameters we can steer is the fraction of ranks involved. If we would like to have random communication partners, and how much data we should actually transfer over the network. So we've taken fairly representative values of about, I think, 10% plus minus 5 for the fraction of ranks and data size to be sent in the order of tens of kilobytes. And so this mini app basically generates a bunch of data in a vector. It sends this data to other ranks, and then it collects data from all the other ranks. It basically just does the receive to the I sense. So basically just loops a bunch of probes until it knows it has collected enough data to proceed. And then we start the next iteration. And depending if we want random partners or not, at every iteration, we determine freshly which ranks we should send our communication out. So we started to do some performance measurements with this. We compared our HPE MPI, which probably has been well tuned by HPE, to our machine. And we used also MBA pitch too. At first, we used the stock version we installed via spec. And here we see basically that at the lower end, for up to 5,000 cores, we're pretty comparable between the two. But then once we had higher core counts, we had some issues with MBA pitch. And so luckily enough, DK Panda came to help with his team, and we got some improvements. Our first approach was basically to look at what's going on. So we fired up HPC Toolkit. We looked a little bit into the traces between these communications. And at some point, we noticed that there's a lot of buffer allocated related stuff going on here. So yeah, we're wondering why this is, why that is, and if it has any impact, if we could resolve it. So as a first step, we basically tried to delay this buffer registration or simplify it. So we generated data into a fresh vector at every iteration to kind of simulate loading data from disk. So at the first step, we moved this buffer allocation outside and then sent always the same memory location in the buffer to other ranks. And so we didn't have to re-register this buffer with MPI. So this gave us some improvement, and luckily enough, we also have a version provided now that has this built in. So we could actually keep our communication and we could keep our mini app as it is. And so here on the bottom right, you kind of see the performance after the fixes. So the orange line is what we saw before with the fixed partners. We have quite the regression up here, and we diverge a lot from HPE MPI. With the new fixed version, at low counts, we're comparable. There's this one outlier here for about, I think that's 256 nodes, where our time is still quite high, but then it flattens out, and for higher core counts, we actually outperform HPE MPI in this application test. So this is not the only thing we do. We also looked at the collective calls. And here, basically, we noticed that HPE uses its own module, its own kernel module for XPMEM. And so we had our core services swap out the kernel module for an allocation to use Craze XPMEM module. And then we also see a reduction in latency in the all-to-all between, for the global data exchange in this first work phase at the higher data sizes that we're actually interested in. Okay, so to summarize, we have a bit of a brain building pipeline that relies very heavily on MPI communications. It's a pretty convoluted pattern. We have some problems when switching MPI implementations, mainly due to the data sizes and the variance in communication partners. And we found MBA Pitch 2 to be a viable alternative in the past and in the present to HPE MPI. I would thank DK Panda again and his team for providing us with assistance to getting the tuning right. So this tuning happens mostly via environment variables. We're kind of afraid a little bit that the setup might be a little bit too dependent on the use case. So as I mentioned, our scenarios, our patterns shift a lot depending on which allocation size we have and which problem size. So we hope to work that out better in the future. Yes, so thanks again. And for future work, we would like to resume this. We would like to eke out this outlier, for example, try to get some better performance than HPE MPI. Then we'd like to jump back from this mini app to our full-blown executions, because we're still building a lot of brain circuits and want to get the speed up. And then at some point in the future, we'd also like to integrate IME burst buffer usage into our application and test that inside out. Okay, With that, just a brief note, if you're interested in neuroscience, we have some opportunities, so don't be afraid to contact us. And I would like to thank you all for listening.
Thanks. Thanks, Matthias. There is one question in the Q&A box.
So, what kind of monitors? I think our MPI expert Pramod might be able to answer that better.
I assume the question is related to the profiling tools.
Yes.
So yeah, typically on our clusters, we are using Tau and also the HPC toolkit quite often. So one of the profiles was from Tau, and Samir yesterday presented that. And the second, timeline traces, we use the HPC toolkit often.
Okay, thanks, Pramod. So I just want to just give a little bit of background. I mean, for the last few weeks, I think several of my team members have been working very closely with Pramod and Matthias. I think thanks to both of you for providing the access and updating some of the modules and all. This is, I think, one of the